My name is Dr. Renee Hirte. I am founder of Dream for the Cure Cancer Research Foundation, and I guess I have a story to tell. So basically, for some reason, I have had a passion and desire to cure cancer since I was in 5th grade. I don't know why, I don't know where that came from, but that was laid on my heart, so I guess I ran with it. By 8th grade, I felt I knew the answer, that if you could teach the immune system to see a tumor as a tumor and not healthy, that would fix the problem. And then I went on to be formally trained as a doctor of pharmacology with a specialty in developing cancer therapeutics. And the reason I did that, as opposed to going and wanting to be in the clinic seeing patients to help them with cancer, is I felt there was no way to help them that I agreed with. And I never wanted to be a doctor who sat behind a desk and told a patient how many years or months rather, they had left to live. I didn't have the heart to do that. And I wanted to make the tools for doctors all around the world that they would never have to do that again either. So by the time I had graduated, I had my first therapeutic that I had a little idea for become a phase 3 clinical trial that led to a 90% cure rate in multiple myeloma cancer patients. And that drug went on to become the first ever FDA-approved therapeutic in immunotherapy to treat cancer. And it paved the way to show this is how we should be doing this. We should be very specifically targeting tumors so that we don't have side effects and that we actually effectively kill them and so they don't ever come back again. So once I had completed that whole process, I knew this is it. This was the proof of concept I needed. We had one target, we made an antibody against it, and it worked. And then I knew there's more than one target, not only on all the different cancers, but on even each tumor itself, that we could get even better at this. And what about the 10% who didn't make it? What marker did they have? What was about them that we could treat so that we had 100%? Because 90% isn't good enough in my book. So then I started to develop the idea of how do we figure out on every single cancer patient who has a tumor, what does their tumor look like, and how can we teach the immune system to see that it's not belonging there and go in to seek and destroy it. Fast forward 10 years, and I finally had all the pieces come together. The final piece that I needed to actually pursue what I had been chasing for such a long time was the ability to actually process and analyze the data that we would be producing. I'm a real believer in using your time wisely, and I didn't find it to be time effective or cost effective. I'd feel bad spending someone's money researching something just to make data that we couldn't understand. So what I needed was a big data science team who knew how to do machine learning and knew how to use really advanced computer science tools to look at the results we were actually going to be producing. And I found them in Silicon Valley, and we started to write a little code. We looked around the whole world for samples we could make the proof of concept on this on, and we found a little set that we did it. And what we found was profound. And we said, we have something here. We just need to test this in the real world. And so we sat around for about 2 months saying, how do you get a tumor sample when you're just some person on the street? Because for the past 10 years, I had not been actually doing formal research. I was dreaming, I was planning, I was looking at this is my dream project and I will not settle for less. I'm not going to sell out and go work for a pharmaceutical company. I'm not even going to go work in academia because I don't believe in anything they're doing so wholeheartedly that I feel like I could wake up in the morning and go do it. And at any rate, 2 months later, my sister calls and says, hey, are you in town and want to get dinner tonight? And I was like, you know, you have never gone and done anything unplanned in your life, so this is a bit odd. But sure, I can drive 2 hours from Sacramento where I'm working today and have dinner with you. No problem. So I did. And on the way to dinner, I was asking her how everything was going. And she said, I have a brain tumor. And my response was, well, thank you for picking the hardest cancer in the world to cure. But you're in luck. Because that computer code I've been working on with my friends that I've been talking about, well, when we tried to decide what cancer do we tackle first, I said, we have a whole set of samples here. They come from different types of cancer, different patients, different sources. How do we most effectively show people that this will work? So I said, let's focus on one cancer. And how do we pick the cancer? Well, let's pick the number of samples in the group that has the highest number, that also has a healthy sample we can look at, so we can see what's different between the tumor and the healthy. One cancer type qualified, that was brain cancer. So I said, you're in luck. We have the code for that. And then I decided, you know what? This is time for it to actually be something real because, my sister's life was on the line and I lost all of my fear. I approached every single expert, everyone and anyone in the field that I knew could do what we needed to actually make this whole lifelong dream into something that was real, that we could actually help people, and at the very least, just help my sister. So I hired a company to actually come into the operating room, properly freeze her samples, handle her samples, so that we could actually have her be patient number one in our real sample trial. And if she was ever the only patient, it would have been enough for me, because at least, then she'd have a shot, because brain cancer is not real easy to beat using traditional methods at this moment in time. So in all of this, she was basically going to see her doctor, and I was being very persistent about no, we're freezing the samples. It's a third party. It's not them, no. And she's like, no, the surgeon says he can freeze it. I'm like, no, it's a very specific protocol. And in the end, she basically said, I want you to come meet the doctor with me the day before the surgery because I know that I need your approval, that he qualifies, and I don't know the right questions to ask, and even when you write them down, I just don't understand. So I said, okay, let me drop everything and fly to Minnesota and meet with your doctor. And 2.5 hours after meeting with this doctor and him asking me, what in the world are you doing? And so I told him, and he said, you know something? It sounds like we should be collaborating. And I said, sure, succeed in this first, and then we'll talk. So a week after the surgery, we met, and after at least 5 hours of meeting and talking about it, we had launched an official clinical trial at the Mayo Clinic. He asked me, would you like to help your sister, or would you like to help a lot of people? I said, let's help everybody. So at any rate, that's where that all started, where now we had real-world samples. So then I needed to chase down the people in the world who could actually process them, and the only guy in the world who actually qualified was in Zurich, Switzerland, at an institution called ETH Zurich, which, if you haven't heard of it yet, just got voted top 5 in the world for institutions. That's where Einstein did his Nobel Prize work, along with 40 other Nobel laureates. So there's pretty high caliber science and technology going on at this institution. And with an extreme amount of persistence, I finally got the head of that lab who had invented this technology that we needed to agree to actually work with us and process all of our samples for us. So I flew to Switzerland with a box of brain tumor samples, which made for an interesting interaction with a flight attendant who insisted we put them in the top bin, and I said no. So they flew next to me in a seat with a seatbelt, because those samples were too precious to ship. My sister was in there. So I ran the first set of samples, which in very short terms, you can look at the surface of a tumor, and basically each one has a signature, a very unique signature of proteins that are being expressed, usually in response to the environment that they're in, which is indicating that they have an issue, which would lead to excessive proliferation and division, which makes a tumor. So basically, we came back with 5 patients' data where we saw their signatures and found 25 unique targets that were on tumor and not on healthy cells. First, I'm going to go into the background of what is it that we do and why does it work, and then talk about how what Evolv has to offer really helps contribute to making it even more effective. So first, I'm going to show you a video that's going to give you a background in the most simple terms I can find in video format in 3 minutes about what is immunotherapy and why is it such an effective way to treat cancer.
When it comes to treating diseases like cancer, modern medicine has an impressive arsenal. And one of its most versatile weapons are Y-shaped proteins called monoclonal antibodies. Our immune systems already produce their own antibodies. They come in billions of variations, each matching a specific target, such as a particular toxin, bacteria, or virus. When they bind to their target, they send a signal. This bacterium is now marked for destruction. These naturally produced antibodies are pretty effective, but in the 1970s, scientists figured out how to mass produce them. They can take any specifically targeted antibody and make huge numbers of identical clones, which means you can produce monoclonal antibodies for almost any disease you can think of. Which brings us on to cancer. Mutated cells that start growing out of control can be deadly. But the body's immune system isn't defenseless. Meet the CTL. It's a type of white blood cell that can detect infected or mutated cells. And when it finds one, it secretes toxic molecules that initiate a self-destruct sequence. But the CTLs do sometimes need to be reined in. If they get over-enthusiastic, the immune system can start causing collateral damage, attacking healthy cells. Fortunately, the CTLs can be turned off by activating a molecule called PD-1. The trouble with having an off switch is that cancer cells can exploit it. Many cancer cells protect themselves with a counter-receptor called PD-L1. This is where monoclonal antibodies come in. Scientists can use one of 2 types. One that binds the PD-1 molecule, or another that goes for PD-L1. Thanks to the monoclonal antibodies, all of the CTLs are fully activated and can blast away the cancer. This is just one way that monoclonal antibodies can be used. They've already proven effective against a whole range of cancers, and yet more are still being developed. With their ability to be targeted at specific diseases, monoclonal antibodies are turning out to be a game changer, and one of our most effective weapons in the fight against cancer.
So that's the first video that people actually started to understand what I was talking about that weren't in the field of science. So basically, in short, antibody therapy is the best technology for cancer therapy that has ever been seen, basically. So to give you an idea of the difference in survival rates and the impact on quality of life patients will have is that if you think about breast cancer as the #1 cancer that ever really had targets that we knew. So now if you hear even today that there's at least 3 markers that a breast cancer patient might have that are very targetable. The survival rates in that are 90% and getting better every day, basically. If you get breast cancer, there's probably a 90% chance you're going to live enough time to have a shot. So to compare that to brain cancer, who has made basically zero advances in survival rates in the past 30 years, they have a 2% chance to make it to 2 years. There's one difference. Breast cancer has immunotherapy available where they can very specifically target these proteins on the breast cancer tumors that are not on the healthy tissues, or at least upregulated so much in the tumor that they serve as very effective targeted therapies, which can be used either in combination with chemotherapy and radiation or on their own, depending on what stage and whatever's going on with the cancer. But basically, that's the difference. So my mission in life was to find targets on every patient there ever was so that they'd all have the same shot and all survive. So here's a basically very simple illustration of how this is working. So you have a tumor cell, and the little shapes on the outside are proteins. So every cell, it has these different things, and proteins are one of the top things that are expressed on their surface. And those are basically signals to other cells and themselves as to what is their job. So some of these proteins have a job of making the cells divide really fast. And those are the ones we're most interested in. And as you can see here, we also have the healthy cell, who shares not the same profile, but similar in some ways. So they share one protein target in common, so we don't want to target that one. So if you target all of the other ones with antibodies, which are shown here, they very specifically go in and bind to the protein on the cell surface, and that does two things. Number one, it turns off the function of the protein itself. So the first target we ever had was a protein called CD38, and that was involved with signaling the cell to divide as fast as it could, activating those pathways. So when you bound to it, it turned off that activity. So the cells could no longer divide so quickly. And it also sent out an alert to the immune system to say, this is a bad cell, come kill it. So no matter where a tumor cell is in the body, if it has these markers, and at no matter what point in time it comes, so even years later, if it comes and it has those markers, the immune system with this kind of therapy is trained to seek and destroy those tumor cells no matter where they're found in the body. So that's the basic concept of what we're doing. And if you can go to the next slide, it basically gives an illustration of exactly what we're doing, where we have a patient. Their tumor cells are extracted either by biopsy or surgery when they remove the tumor. Certain institutions, very few though, like the Mayo Clinic, they freeze the tumors upon removal no matter what, and they save them so that they can have research done on them in the future if the opportunity comes about. And then in step 2, we basically send the samples to Switzerland at this point in time, and we separate the cell surface proteins from the cells themselves. And then we identify the proteins that are only on the tumor cells and not on the healthy cells. With that, we can engineer antibodies against the proteins that are just on the tumor. And then those antibodies are given to the patient. And then cancer cells are targeted and destroyed everywhere in the body. This is a very effective strategy. So the really cool news about the brain cancer trial that we started at the Mayo Clinic was when we found these 25 targets that were shared on basically the entire patient population. There were a handful that were differentially expressed. So in that, there were actually 4 groups of patients. They all shared, every single one of them, at least 16 of the proteins. And then the other ones varied from patient to patient. So if you treat with the wrong immunotherapy, it will not work, because if the patient has not got the target, it will not find the tumor cell. So we got really lucky in this one, because out of those 25 targets, there's 5 that already have FDA-approved drugs against them that are effective in other cancers. So that means that we can actually test those in the brain cancer patients to see which one will work best, because in theory, it should work on any tumor that shares that target. So that's where we're at right now, is in the stage of determining which of the 5 are actually most effective at treating someone with a brain tumor. Interestingly enough, the first therapeutic I ever made is also a target in these patients, all of them. So that was kind of a full circle thing to happen, which was nice. So now you're probably wondering, what in the world does this have to do with Evolv? And I'll tell you that when you're doing this kind of therapy, you need to have a functioning immune system. And a lot of cancer patients, one of the biggest challenges is that their immune system is compromised, either by treatment or by the disease or whatever it is, but they have these compromised immune systems. And even if they don't have a compromised immune system, in order to have something like immunotherapy work, you actually need to have, ideally, the most optimal functioning immune system that there is. So when I found out about Ace Mannin and what it had been doing and heard some of the stories and then did a whole bunch of research and due diligence, because no matter what it is, I'm going to suggest my sister to do it, better be right. And I figured out this is good. So at any rate, Immune does an excellent job as an integrative health support strategy. For any person with cancer that comes to me, I actually now tell them that Immune is one of the first things they need to take. You have a great set of products that will do tremendous help, not only on their own, but in the future with the combination of immunotherapy. And one of the most exciting things is that one of the things Sam has offered to do is actually help be able to implement Ace Mannin and Entourage in the clinical trials at the Mayo Clinic. So not only will we test those 5 FDA-approved drugs to see which one's most effective, but we will also have Entourage and the Immune in the sets of patients to show that it's even more effective if you add those, because that is what I predict is going to happen.